Welcome to Dr. Balga's podcast for the Curious Doc. Today is December 19, 2025. This video is on a Nature article published online on 2 days ago, 17 December 2025, titled Transient Hepatic Reconstitution of Trophic Factors Enhanced Aged Immunity. There's an accompanying news article by Heidi Ledford in the same issue of Nature titled Restoring Youth to Old Immune Cells. mRNA therapy turns back the clock. Testing in mice suggests that rejuvenating T cells could make vaccines and cancer therapies more effective. Aging leads to thymic involution, loss of naive T cells, reduced T cell receptor diversity, and impaired vaccine and tumor immunity. So the hypothesis was the liver can be repurposed as a transient factory to restore age-diminished immune trophic signals. The key pathways targeted were Notch pathway, FLT3 ligands, and interleukin-7, IL-7. The model was age C57BL by /6 mice, aged around 72 weeks, compared with young adult mice of 6 weeks. Intervention: lipid nanoparticle encapsulated mRNA encoding the Notch gene, the FLT3 ligand, and IL-7 were dosed twice weekly for 28 days. Sample sizes typically 4 to 12 mice per group depending on the assay. Multi-omic profiling included 97,000 blood T cells and about 1.26 million thymic spatial positions. They used two-tailed statistic student t-tests, 1 or 2A ANOVA with post-hoc testing, and log rank test for survival. What did they find? The combination DFI expanded common lymphoid progenitors and restored thymopoiesis without altering hematopoietic stem cell composition. Naive CD4 and CD8 T cells increased significantly. Naive to memory ratios improved, p value less than 0.001 in multiple assays. Vaccine response: approximately two-fold increase in antigen-specific CD8 T cells in aged mice. Cancer models: improved tumor control and survival. 40% complete tumor rejection in aggressive melanoma model. 40%. Safety: no increase in autoimmunity or in different models. Reversibility: immune enhancement waned after cessation of mRNA dosing. Limitation of the study: it's mouse-only data. Long-term safety of repeated dosing in aged hosts remains unknown. Key takeaway: transient liver-directed mRNA therapy can rejuvenate aged immunity without breaking tolerance. The News and Views article accompanying was titled Restoring Youth to Old Immune Cells. The problem: aging reduces both quantity and quality of T lymphocytes, weakening efficacy of vaccines and cancer immunotherapy in older adults. This approach of twice weekly liver-targeted cocktail of 3 messenger RNA encoding IL-7, FLT3 ligand, and DLL1 rejuvenated T cell biology in aged mice. Mechanism: the liver acts as a transient factory, restoring thymic signals without directly repairing the involuted thymus. The key findings: treated mice produce more naive T cells and showed stronger vaccine responses and improved tumor control. Reversibility: immune benefits waned after therapy was stopped, suggesting controlled and non-permanent immune modulation. The big picture: T cell aging is modifiable, opening a path to healthier immune aging and improved late life resilience. Clinical pearls: immune aging is not irreversible. The system retains latent plasticity. Transient signaling restoration avoids chronic cytokine toxicity. mRNA delivery enables spatial and temporal control of immune modulation. The strategy is disease agnostic and synergizes with vaccines and immunotherapy. This is a conceptual shift. Treat immunoaging as a reversible systems failure, not as an inevitable decline. These are some eponyms related to this study. Hit pause and read at your leisure. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.